Okay. Hello. So I will talk about label-free quantification. So after Jung was showing the newest research, this is a little bit older, some years, but it's very well established. And so let's just start. First, I want to show you this table, which could be the output of an experiment. So here you have some samples that you run, 5 samples in the columns, and the rows, these are proteins from 1 to 5. And these numbers in the cells, they indicate intensities of these proteins measured in all these runs. So now you could think of what can I do with this data? And first you can say, okay, I want to compare proteins across the samples, like one protein, for example, protein 2, across all these samples. This would be a relative quantification. But you could also say, okay, I'm more interested in comparing the quantity of proteins within one sample, or even get a absolute value, like copy cells per number, numbers per cell, or milligrams or something like this. For the relative quantification, I will talk about the MaxLFQ algorithm, and for the absolute quantification, I will discuss the proteomic ruler. So let's start with the relative quantification. So actually there is 3 possible ways to do it. So you can do a classical labeling. Can you see it? Yeah. Classical labeling, like MS1 labeling using SciLec, or even MS2 labeling with TMT, which is very popular. But there's also label-free, and label-free has some advantages. So it's very easy to prepare, so you don't have to put the labels on your peptides. You have a higher dynamic range. There is no limit in the number of samples. If you just look at this plot for SciLec, there you only have 2 or 3 channels in label-free. There is no limitation. You also compare chemically identical peptides. So, I mean, these labelings should not affect the peptides that much, but still it's possible that you have like slight shifts in the retention time, for example. And there are not only advantages, there are also like some challenges that have to be tackled and that I want to show you now. So let's now have a look at the SciLec labeling. So this is not working anymore. Let's try the red one. Okay. So here we see a SciLec pair, which, like, let's assume like one peptide was identified, and then we want to get a ratio between these 2 peptides to see how they differ. And it's actually quite easy to find the SciLec partner, right? It's just a difference of around 8 Daltons. And the retention time is basically the same, and they're in the same sample, so everybody is good. Everything is good. That's not so challenging, but if you now go to label-free, you compare 2 different runs. So across these runs, and you want to compare one peptide in the first sample with the same peptide in the other sample, which is not that easy as it looks on this figure. There are several reasons. Petra already talked about that. So the elution time differs. So the chromatography is not completely reproducible. You will have slight or big shifts depending on your column and your LC. But if you have, sorry, like the pointer's not working. Mouse. Okay. So here you can see, like, this is the isotope pattern in sample A, and here it is in sample B, and there is an offset in the retention time. And there's also another problem is the acquisition, like the DDA procedure, is also not completely reproducible. So, like, always there's an MS1 scan, and like the top peaks are selected for fragmentation, but this changes, so it's not always the same peptides. And here you can, as you can see here, so if there is a cross, it was not identified in sample A, but in sample B it was identified. So it's actually not so easy to match these two. There are some, like, it's not completely overlapping the identifications. And also, if you do prefractionation, the peptides, they can appear in several fractions. Okay, so let's start with the illusion time, let's call it problem. So on this plot, you see two CMS runs, the retention times of the identified peptides plotted against each other. And you can see like a very dense region, which is also changing with the gradient, and there is a retention time difference, like starting from 1 minute going up to like 2.5 something, and but there's also like less dense regions, and yeah, so exactly. So if you now take the like the most dense regions or over the retention time, there can be this recalibration done, or like it's called the alignment of retention times between two samples. And once this nonlinear function is determined, you can just project it down so that there's like, you just align that dynamically. So here we see a correlation plot. This would be like an ideal result of this alignment, right? So like every peptide is eluting nearly at the same time in the two samples. And this can be done on a large scale over many samples. Okay, so now we have aligned our samples, right? Peptides in this new time scale elute at the same time. So let's have a look at the next problem, which is the sequencing, the MS-MS sequencing. So again, here we see our alignment, whoop, 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 and then this is the problem. We have an identification on one side, but here there is no peptide identified. So how should we compare? What should we compare to each other? And the solution is the matching between runs, which basically looks on the MS1 level in the aligned LC runs, if there is a isotopic pattern which matches, so which is eluting at the time that we expect it to elute, and if it has like the mass that we expect in the charge state. And this method is actually quite helpful because first it was a disadvantage that we have sequencing which is not completely the same, right? But now we make we just use all of them and match between these between the runs, and then you can boost the match, like the possible matching, by a factor of 2 or so. Okay, so so the first problems are solved, right? So we found our peptides in both samples at the retention times, and we could now compare them. But then we get the next problem, and for this I want to show you these two red guys, and if we now look at them and say, okay, let's compare the size or the height of them, probably everybody would say, okay, like the right person looks a little bit taller or much taller than the left person. But if I show you another picture of those guys, which is probably taken like from a slightly at a different day, slightly different perspective, let's say different LCMS run, right? And we also take into account that they're not alone, right? So there's like all this crowd around them. Then you would say, okay, they are like taller than the rest of the people, but probably like they're probably like they have the same size, right? So you're normalizing that and take into account the fact that these are different measurements. Okay, so the peptide intensities somehow have to be normalized. Awesome. Okay, so now let's have a look at this experiment. So we have samples from A to C, and there is fractions, and we have a look at fraction 5 to 8, and there is peptide P, which is yellow. And this peptide P appears for sample A in 3 fractions, right? And like the circle size is the intensity in all of these fractions. So if we now want to calculate an intensity for the whole sample, right, over the fractions, the first idea would be to say, okay, like we just take the intensities and sum them up. But as we already learned, okay, like the intensity can probably change between these samples just because of the measurement, not because of something else. So we would need to multiply some normalization coefficients, which are called n. So just to explain it, these numbers are just indices, right? It's not the power. So, but there's so many indices, I put them there. And so these coefficients, they exist for every LCMS run. So we have this table again, and then we have, for every run, we have a normalization factor, which is multiplied to the intensities. And then we can continue. So the peptide P is also present in the sample B, but in other fractions, and also in sample C in other fractions. So only in 2 in this case. Okay, perfect. So we could calculate our intensity now for peptide P, and we could also do it for like a peptide Q, which is green. And we would use, of course, the same normalization factors as for peptide P, which we still don't know, right? So this is like, it's always nice when you have a formula, but you have to solve it. Okay, so that's the problem. So we know how to calculate these peptide intensities, but they all depend on these normalization factors n, which is like a set or a matrix, however you want to call this. And the solution is that a function is defined which depends on these normalization factors and which takes the squared logarithms of the ratios between the peptides in the different samples. And this is then all these functions are summed up for all the peptides that were matched, and then this is minimized. So what does it mean? Actually, the minimum of the logarithm would be 1. So if IPA and IPB are equal, then that's the minimum. Huh? And we do it over all the peptides. So actually we try to like get the same intensities over all of the samples for all the peptides. Okay? So formulas are always a little bit difficult, so we can also have a look at a nice figure. So these are our two runs before and before the normalization. So here we have like a higher overall intensity than here. And the normalization, okay, we cannot compare that. We already know that. So then we take our normalization factors and apply them to all the peptide intensities, and then everything is comparable. Okay. Let's go back to the formulas. So if you have many, many samples, right, this HP function becomes very complicated because you have all these sample pairs, and this is a function, a squared function. So this becomes very computationally expensive, as we say, and then your computer gets slow. So if you have many, many samples, you have so many comparisons, like it just slows down. And there is a very nice solution, or like people call it a trick, which is only take a subset of these pairwise comparisons, because like there is so much data that is compared, and like you don't have to compare all the peptides over all samples. So just taking a subset is also doing the job perfectly, and this is called fast LFQ. Okay. I think now we're at a point where we could compare our two samples, or like not two samples, but thousands of samples with fractionation. So there can also be a problem. So if your samples are too different, which is indicated here by the colors. So if, for example, like it can happen that in your measurements, like one LCMS1 was just empty or so it's not comparable, then the normalization fails, and you get an error message. So it doesn't necessarily mean that MaxQuant has a bug. It probably means that something is wrong with your data, or something is wrong with your experimental design. Yes?
A very quick question. How is the normalization at peptide level impacted by the missing data?
By the missing data?
Right.
Since, I mean, there's only like the peptides intensities taken into account, so
but when you're
missing data is like
right.
not existing at the peptide level, right? I mean, so there's no missing data, I would say, right?
Maybe I'm missing something. We'll talk offline. Yeah.
Maybe just to clarify, so because we're restricting ourselves exactly only on these peptides, where in the pairwise comparison both are there, we avoid the missing value problem, basically.
Okay, so if you get this error message, just try to check your data, try to check your experimental design, and the rest of the analysis before you go to the Google group and say, I found a bug in MaxQuant. Okay, but otherwise, you should be able to normalize your data, and then you could proceed, or like the algorithm could proceed to the relative protein quantification, because we are still on the peptide level, right? So let's have a look at this nice protein, which has some identified peptides, P1 to P6. And these peptides were measured in our samples from A to F. So we can use this table now to calculate ratios by taking the ratios of the peptides between two between two samples, right? So, for example, ratio AB is it's the median actually of this as the ratio between A and B for peptide P and for peptide P6, which are both present in both samples. And doing this, all the ratios between the samples can be calculated if there is enough data to do that, right? So there is a threshold which is called the LFQ minimum ratio count. So if you want to calculate the ratio between E and F, for example, which is this one, there's only like one peptide which was in both samples, and therefore the ratio is not calculated. Okay, so now we have ratios for every peptide, no, for protein, sorry, for our protein between the samples. And using these ratios, intensities can be calculated by just saying, okay, like the intensities for the proteins are also the same as calculated in this table. And if there is not enough data, like the intensity is 0. So here we now see we get an intensity profile over the different samples for our protein. Very nice. But like there's still this sample F, right? So there's a 0, and probably like this can be rescued, this value. And there is a possibility which is called stabilized large ratios. So in this case, the overall intensities of the peptides in every sample are summed up, and the ratio of this is taken. And then you can still get a value, which is probably not that accurate as the normally as the other one. Okay, so now I think we are done. We can compare our intensities, and let's do some benchmarking. Let's have first a look at the small ratios. So for this, we have a look at this experiments, which is HeLa and E. coli mix, 1:1 or 1:3 and 24 fractions and 3 replicates. And so in the paper, in the LFQ paper, the MaxLFQ was compared to spectral counting, which is a like kind of old-fashioned way, and also the summed intensities. And as you can see, the spectral counting cannot really good separate the two species. For the two samples, the summed intensities are already much better, but the MaxLFQ it looks a little bit better in this case. And if you go to the histograms now, which is probably like easier to interpret, you see that this is on the log scale, that the distance here is nearly to what we expect from the experimental design. Okay, so it looks that this method is usable for small ratios, and this brings us to testing or benchmarking with large ratios. The experiment in this case is E. coli with spiked in UPS standard, like UPS 1 and UPS 2, which is a commercially available standard of human proteins, 48 proteins, and that's basically used to quality control the dynamic range. So in the UPS 1, they are diluted, so the abundance of 8 proteins is always like by a factor of 10 reduced. And On this plot, you can see these clusters of 8 proteins each, and you can see how it really, the MaxLFQ is able to go down or to quantify to very, very, very small or large ratios, however you calculate it. Okay. This shows also a comparison for our large ratios for the MaxLFQ against the spectral counting again, and the spectral counting is actually pretty bad. What's also noticeable is that like the small ratios, right, they are completely missing. This is because the method of spectral counting is just counting the MS-MS events, so only the high abundant peptides are in this data anyway, and in the matching between runs and all the strategy makes it now possible to go very, very to very small intensities, what you can already see in the summed intensities, and the MaxLFQ with this very nice way of calculating the intensities is pretty close to the diagonal of this correlation plot. Okay, so what if there's missing values, but not for the normalization? So in this, like, this is just showing how many missing values can appear. So if you have missing values, you can either like drop them or you can try to rescue them and have more statistics. And in this case, it's important to know a little bit why values are missing, and this is because the intensity is low. So the solution in statistics is called imputation. So actually the values, the missing values are just values that are taken out of this distribution. Okay. So let's now switch to the absolute quantification. Up to now we compared proteins, like the same proteins over several samples, and now we want to get absolute values, such as copy numbers per cell or milligrams. And the problem here is, I mean, it's still the same problem, right? It's not inherently quantitative. So the peptides behave different. All the peptides behave different in this mass spectrometer, and even the runs are not reproducible. So this means, I mean, the MS intensity is strongly correlated with the input amount, but it's not so easy to compare. So you have to correct for the protein size. So it really matters. Your MS signal from a protein is dependent on how your protein is made up. So how flyable are the peptides? So this has to be corrected. And it's also still there is no unit, right? So the mass spec doesn't tell you, okay, like we have such much grams or whatever. So an absolute scale is needed. And this can be done using the proteomic ruler. So here on the left, we see the MS signals for all proteins now. And on the bottom, there is the histone MS signal, right? And for the histone, it is known that the mass of the histone is more or less equal to the DNA mass in a cell. And the DNA mass can be calculated easily. So if we now know, okay, if we have, like, for example, this protein, we just take the ratio to the histone, total histone MS signal, and then out of that we can calculate the protein amount in the cell. So this is our absolute scale. And to benchmark this, so-called pressed proteins were spiked in, like silac labels. This is also a possibility to do an absolute quantification, but it's a lot of effort to do it. But it's a very good validation. And here you see the quantification, so in copy numbers per cell, the pressed silex experiment, which is supposed to be very accurate, against the proteomic ruler, which just is like very easy to handle. And I mean, these data, they just more or less are for free with your label-free quantification. And it's surprisingly well performing, so there is not a big difference between these two methods for quantification, absolute quantification. And on the right, it was also compared to classical methods like cell counting or DNA determination. Okay, so now we discovered the dimensions of protein quantification. So relative quantification using the MaxLFQ and the absolute quantification with the proteomic ruler. So thank you, and I'm happy to take questions.